If we look at carbon and we do some biochemistry, we'll see that it can form up to 4 covalent bonds. We can see this in methane, CH4. Okay, it's formed 4 covalent bonds with the hydrogens. Okay, a covalent bond is essentially when you share electrons, as you can see here, between nonmetals. Carbon is not a metal, and neither is hydrogen. So this is why it's a covalent bond. These covalent bonds between the carbons and the hydrogens are going to be the building blocks, the main building blocks of certain chemicals, okay, that we see in biology. Okay, so carbon-carbon covalent bonds are strong and stable, and this is why, I repeat, they are the building blocks of most biological molecules. And this is the case the longer the chain of carbon-carbon bonds is, the more strong and stable it's going to be. Now, this, for example, is a fatty acid. There's going to be a hydrocarbon chain, okay, hydrocarbon, and it's a chain, as you can see here, and it's going to end with a double bond between a carbon and an oxygen, and a bond with a hydroxide, okay, an OH. Now, these carbon-hydrogen bonds, or carbon-carbon bonds, can also be in ring form, as you can see here, glucose. So they can either be a chain or a ring. Looking deeper into glucose, we're going to see the following. Okay, so if you have 10,000 glucoses, okay, this can give you something like this, which is glycogen, a branched polymer. A polymer is made of monomers. In this case, glucose is the monomer. Monomer for one, poly for many. So many monomers makes a polymer. We know that glycogen is the storage form of glucose. So you accumulate a lot of glucoses, they bond with each other and form the polymer, the glycogen. Moving on to macromolecules, okay? We'll see that there are 4 main types: nucleic acids, proteins, lipids, and carbohydrates. Okay, if we're looking at nutrition, we're only going to focus on these 3, okay? If you do sports science, you'll know that. Now, these macromolecules are going to be made of monomers. I've spoken about these before, because these make up the polymers. But how does this happen? Well, this is done by either condensation, polymerization, or condensation reactions. Now, condensation polymerization applies to carbohydrates, proteins, and nucleic acids. Now, condensation reactions, those are for lipids only. Okay. Now, let's have a look at condensation polymerization and using glucose as an example. Okay, so this is going to be this reaction between 2 glucose molecules. I repeat that condensation polymerization happens in all macromolecules except lipids. Now, the first thing that's going to happen is that one of the glucoses, okay, I've chosen alpha glucoses for this. I'm going to explain what the differences between alpha and beta glucoses is later, so don't worry about that. So one of them is going to lose a hydrogen, and the other is going to lose a hydroxyl, or an HO or OH, okay? Now, if there's a H, if there's 2 H's and an O, this makes up H2O, which is water, okay? So remember that in condensation reactions, a water molecule is always lost. In hydrolysis reactions, which we'll look at later, a water molecule is gained. Now, what happens now? They've lost these components. An oxygen is going to join the 2 molecules together, okay? And when 2 glucoses are joined together, we get maltose, a disaccharide. Glucose is a monosaccharide, 1. If you mix 2 glucoses together, you get a disaccharide, di meaning 2. Okay, that's not the greatest 2. I guess that's better. Whatever. Now, maltose is an example of a disaccharide. This bond right here, this oxygen that joins them, these 2 together, is a covalent 1,4 glycosidic bond. Okay, glyco for glycogen, because as we'll see, when more than two glucoses join together, we get a polysaccharide such as glycogen. Okay, so remember that poly is two or more, and maltose is the di 2. Okay. Now, why is it a 1,4 glycosidic bond? This is because it bonds between the carbon 1 of them and the carbon 4 of them. Now, this would be the carbon 4 of this molecule, and this would be the carbon 1 of this molecule, if that makes sense. Any questions can go in the comments, as always. Now, hydrolysis. This is the opposite of this reaction here, of condensation. Water, this water right here, is going to break the 1,4 glycosidic bond, okay? And the polymer, or the disaccharide as well, can be divided back into the monomers for energy, okay? So this is when we break down glycogen into glucose for energy. This is glycogenolysis. Hydrolysis is going to happen. Remember, monosaccharide, 1. Disaccharide, 2 of these. Polysaccharide, 2 or more of these. Now, monosaccharides are what we use for energy. Let's look at different examples. The one we've already looked at, glucose. Just note down that glucose has isomers, okay? An isomer is essentially a molecule which is the same. It contains the same components. The only difference is that, for example, the OH might be at the top or at the bottom. And this is how we differentiate alpha glucose and beta glucoses, okay? Now, in alpha glucose, this hydroxyl group is at the bottom, connected to the carbon 1, but it's at the bottom. In beta glucoses, this hydroxyl is above. Okay, this is very important to note this down. Now, these two different types of glucoses, these isomers of glucose, cause variety in polysaccharides. Okay. Fructose is another monosaccharide, also used for energy. It's mainly found in fruits. You might know this. Galactose. This is found in milk, for example. Now, monosaccharides have a classified number of carbon atoms. So, pentose will have 5 carbon atoms. This is indicated by the pent. Remember, pentagon, 5 sides, pentose, 5 carbon atoms. Let's have a look at the properties of glucose. First property, glucose can dissolve in H2O, in water. It is soluble in water. Why? This is due to its polarity. If you haven't seen my video on water, I believe it's A1.1. Check it out so you understand polarity. So this is because of its polarity from many hydroxyls, hydrogen bonding with the water. Okay, so these hydroxyls form hydrogen bonds with water. So these two things are going to cause the solubility of glucose in water. Now, why is glucose polar? Well, this is because its oxygen has a partially negative charge. This oxygen right here, okay, you might want to pause and zoom in. So glucose always has this oxygen, right? And it has a partially negative charge, and this makes the molecule polar, the monosaccharide polar. I repeat, I suggest you look at my video on A1.1 if you want to understand polarity better. Now, because it's soluble in water, it can dissolve in blood plasma, and so we can carry it as energy. Now, in water, have this in mind, glucose can enter what's called an open chain state. This is where carbon 1, right here, is going to rotate. And because it rotates, the position of this hydroxyl here is going to change a lot. So alpha or beta are randomly assigned to the glucose. Okay. So also have in mind that when this glucose is in an open chain state, it's simply glucose. It's not alpha or beta yet. the rotation of carbon 1 is going to decide. Another property. So glucose is stable. What does this mean? It is a cyclic molecule. Now, as most of its hydroxyls are in equatorial positions, okay, equatorial meaning around the equators of the molecule, this provides the structure for the following polysaccharides: cellulose in plants, glycogen in plants and animals, and starch also in plants and animals, mostly in plants. Now, third property, glucose can be oxidized. Oxidization is basically losing electrons. Okay, and it loses its electrons during our aerobic respiration, which has the aim of producing ATP for energy. Okay. Wow, this requires a lot of knowledge from other topics. Damn. Anyways, starch is the main store of energy for plants. For humans, it's glycogen. Okay. Now, starch can be divided into two types: amylose and amylopectin. We can see the different shapes of these. We'll see that amylose has a more coiled shape, and amylopectin is formed more like a tree with branches. Okay. Now, this is called a compact helix shape, and it makes it a better store of starch in the long term. Okay. Now, the branches of amylopectin allow efficient storage and release. Okay. So efficient storage because of the branches we can fit the starch into tighter spaces, okay, compared to the compact helix shape. And the release is also faster because there's more surface area, okay, to then break it down and transport it through the fluid, okay. Now it is less efficient long term as it takes up more space. Now both of these have alpha 1,4 glycosidic bonds, okay, and both are insoluble in water, and this maintains osmotic balance of the cell. Let me just underline that. Osmotic meaning osmosis, so movement of water from a high water potential to a low water potential. Now, amylopectin has occasionally certain alpha 1,6 glycosidic bonds, and these are the ones that are responsible for the branches that we can see around here. Now, as I said before, glycogen is the main store of energy for animals, okay? It's broken down into glucose, okay? And this is done by liver cells. Now, glycogen is stored both in muscles for energy and the liver, where liver cells convert it to glucose. Now, it has alpha 1,4 glycosidic bonds that form the linear backbone and alpha 1,6 glycosidic bonds that create the branches we can see here, just like in amylopectin. Now, these branches, again, they cause a more efficient storage and release of glucose due to the higher surface area, just like in amylopectin. And we can see that it's relatively insoluble in water, and this also helps maintain osmotic balance in our cells. Cellulose is what makes up plant cell walls. Let's have a look at it. Now, it is composed of unbranched chains grouped into bundles called microfibrils. You can see these branches of polysaccharides are not branched. There's no branches, they're just straight. And these three together make up a microfibril. A group of microfibrils makes up a fiber. Probably heard of that. Now, microfibrils are held together by hydrogen bonding. So the bonds right here, these bonds are hydrogen bonds. Okay? And they occur between adjacent cellulose molecules. Adjacent meaning like this. Bonds between these, again, are hydrogen bonds. The bonds between them, so these are the vertical bonds, right? The horizontal bonds here, remember, these are the glycosidic 1,4 bonds, okay? Now, carrying on, I just need to, okay, this gives, so these hydrogen bonds right here give the cellulose a lot of tensile strength. And this is a very important function as a cell wall. Now, beta glucose molecules alternate in orientation. So we can see all of these are beta glucoses, I believe. And here the glycosidic bonds is at the bottom, here it's at the top, here at the bottom, here's at the top. That's why we say that they alternate in orientation. Types of lipids or fats. Now, first have in mind that lipids in general have low solubility in aqueous solutions, okay, such as water. So they're not going to easily dissolve in water. And this is due to their hydrophobic nature. If they were hydrophilic, they would be more likely to dissolve in water. Now, hydrophobic molecules are non-polar and hence can dissolve in non-polar solvents. Now, water is polar, so again, this is why lipids cannot really dissolve in water. That makes sense. Now, let's look at different examples. Okay. By the way, this section right here, if you watch the video on A1.1 water, that's going to help a lot in the understanding of this. So it interlinks with that topic. Next, first type of lipid, steroids. Okay. These are composed of 4 carbon rings. We saw that carbon can be in rings before, and these are fused together. An example of this can be testosterone or estrogen, progesterone, okay, hormones like that. Those are the easiest examples. Waxes, okay, another type of lipid. These are completely water insoluble. They cannot dissolve in water, and they have a high melting point. An example of this is on the surface of leaves. You can see certain waxes forming a waterproof layer that reduces the rate of transpiration. This in the plant topics. If you haven't done them yet, that's going to help in your understanding of this. Triglycerides, another type of lipid. These are consumed in foods and they are synthesized, okay, or created by the liver. So both, they can be consumed and synthesized. Now, they are formed by the condensation reaction between a glycerol molecule, 3 fatty acids, and they are bonded by an ester bond. So here we can see the fatty acid is this hydrocarbon chain, and it bonds with the glycerol right here. And this is done by a condensation reaction, where, remember, condensation reactions, a water molecule is lost. Now this up here is when we look at it for only one fatty acid and the glycerol. So 3 of these are these 3, okay? And this is how it would look like, the complete version, the complete bond between the glycerol and the 3 fatty acid chains, okay? This is only one fatty acid chain. And as you can see, the ester bonds are right here. Last type, phospholipids, okay? Phospholipid bilayer, another topic that interlinks with this. Now, this is essentially similar to a triglyceride. It's just that this time it is a glycerol molecule modified with a phosphate group, this right here, phosphate group, and 2 fatty acids, not 3, but 2. These 2 right here, 2 fatty acid chains, bonded by ester bonds again, and then the modified glycerol. Let's have a deeper look into phospholipid. I just got a better microphone because I realized how bad the audio was before. Anyways, so phospholipids, let me give a clear explanation of this. So they form by a condensation reaction. Remember, water is released. Now this is the glycerol, this whole thing right here, okay, including this hydrogen. Now it is modified with a, as you can see, phosphate group, okay? Now, how does it bond to the 2 fatty acid chains? Well, there's 2 ester bonds, one for each chain. And this is basically what makes up a phospholipid. Moving on to the three types of, listen up, fatty acid chains, okay? So we know that these fatty acid chains are hydrocarbon chains. Now, the carbon atoms in these chains are bonded together by single covalent bonds, okay? Now, each atom is also bonded to a hydrogen, as we know. Now, when there's more carbon double bonds, the hydrocarbon is going to have, the fatty acid is going to have a lower melting point, and hence it's more likely going to be a liquid. And this is important when we look at these three types of fatty acids, okay? So they are mainly bonded together, the carbons, by single covalent bonds, but we can have double carbon-carbon bonds, okay? So the single covalent bonds would look like this, okay? Not the best drawing. And the double bonds will look like this right here. Now, starting off with saturated fats. These fats have no double carbon-carbon bonds in their hydrocarbon chains, okay? And this is why they have a linear shape. As you can see here, linear shape. We're going to look into depth into these structures in a second. And they're also solid at room temperature, okay? We know that eggs are solid at room temperature. The fats in red meats are solid at room temperature, okay? And why is this? Well, this is because the less double carbon-carbon bonds, the higher the melting point, so the opposite of this right here. Now, monounsaturated fatty acids, these have one of these bonds in their hydrocarbon chain, and this causes a kink, also known as the bend, in their chain, this right here. Again, we're going to look at into depth in a second. Now, this makes it difficult for the molecules to pack together tightly, and so they have a liquid state at room temperature. An example of this is the monounsaturated fatty acids in olive oil. Polyunsaturated fatty acids, these have two or more double carbon-carbon bonds. Okay, remember, mono is always for one and poly is for two or more. Now, these have multiple of these kinks or bends in their chain, and this makes it even more difficult for the molecules to pack together, and so they have a liquid state at room temperature, and they have an even lower melting point. An example of this, seed oils such as sunflower oil. Not very healthy. Anyways, let's have a look at their structure individually. So, as I said before, saturated fatty acids have no double carbon-carbon bonds, and that's why the structure is so linear and there's no kinks slash bends in it, okay? Now, unsaturated, okay, can be trans or cis, apart from poly or mono, okay? Now let me delete this, okay. In trans, unsaturated fatty acids, the hydrogen atoms are in opposite sides, okay? Remember, look at this, okay? The bottom right, the hydrogens are the blue circles, the oxygens are the red ones, and the carbons are the white ones, right? So the hydrogen here is on the opposite side of the hydrogen here, and this is trans unsaturated fatty acids. Now for cis unsaturated fatty acids, this time the hydrogen atoms are at the same side. Boom. And this causes an even more bent configuration. Simple as that. Now again, we just looked at these. So monounsaturated fatty acids, for example, oleic acid, you can see that there is one double carbon bond, mono, and that this causes a kink or bend like this. Polyunsaturated, two or more. In this case, we can see it has two, and so they are multiple kinks or bends. That line was unnecessary. An example of this would be linolenic acid, an omega-3 fatty acid. Endotherms. These are animals that rely on what's called metabolic or chemical reactions to generate heat, maintaining a constant internal body temperature. Okay. Now, fat or the lipids that we've just seen are stored in adipocytes as liquid droplets that can be broken down into ATP or adenosine triphosphate. Okay, energy. Now, triglycerides that we've just seen, okay, type of lipid, type of fat, also serves as an effective thermal insulator in many organisms, so it helps us maintain our body temperature in cold situations. An example of a triglyceride is blubber, okay. Again, it serves as a thermal insulator, and it is composed of adipose tissues containing large amounts of triglycerides. Now, different animals with this blubber: whales and seals. Phospholipids can be seen in the bilayers of our cell membranes. This links to another topic, okay? Now, these consist of a hydrophilic polar head, okay? So it's polar and hydrophilic, which means it can dissolve in water. It can interact with water, dissolve in water. Now, remember, yes, water is also polar, and that's why. Now we can see the two tails, where the orange table, if labeled here, is straight and saturated. Remember that saturated fatty acids have no carbon double bonds, and so they are fully straight. And we can see the unsaturated fatty acid is going to have the kink or the bend because it has at least one carbon double bond, okay? And both of these tails are hydrophobic, so they will not interact with water, and non-polar, so they can't dissolve in water. Now, because they have both hydrophobic and hydrophilic regions, the head being hydrophilic and the tails being hydrophobic, these phospholipids are what we call amphipathic. If we look at a cell membrane, we're going to see that there's going to be an intracellular fluid, which is the cytoplasm. Okay, so this side at the bottom is the cytoplasm. Okay, and if our cell is in aqueous solution or water, the extracellular fluid is going to be here. Okay, now certain molecules can pass through our cell membranes. Okay, and steroids, okay, a type of fat we've seen before, for example testosterone, are hydrophobic and non-polar, which means they can pass via the bilayer. Okay, so if a molecule is hydrophobic and non-polar, it can pass. Now it's going to pass. Okay, moving on to amino acids. These are monomers, okay, that make up proteins. Mono for one, okay, mono is always one. Monosaccharides, monounsaturated, one. Now these amino acids are made up of the following. In the center we're going to have an alpha, this means alpha carbon, an alpha carbon atom, okay? Now, what we're going to see, there's going to be a hydrogen up top and an R group at the bottom, okay? This R group is going to vary for each of the 20 amino acid types that we have, okay? Some of which are essential and non-essential. We're going to check that out in a second. Now to the left, we're going to see the amino group made up of a nitrogen and two hydrogens, so NH2. And at the right, we have a carbon, again, okay? It's going to form a double bond with the oxygen, and then a bond with an oxygen, which is bonded to a hydrogen, and this whole thing right here is the carboxyl group, okay? Now we're going to look at how the amino acids get together. Amino acids get together via a condensation reaction, remember, releasing water. Now, as you can see from that animation, a water molecule has just been released, and we are left with the following, okay? So we can see these are the alpha carbons, alpha carbon here, and then the carboxyl group of this amino acid lost, I believe, a hydrogen and an oxygen, the HO, and here this amino group lost the hydrogen, okay? Because there would usually be a hydrogen here, and there would usually be a hydrogen here as well, and here there would usually be an oxygen, I believe. Now, this is essentially what's going to happen. A condensation reaction releasing the water molecule. Now this forms a dipeptide, di for 2, and peptide, okay? Okay, this is the bond, okay? I forgot these animations. The peptide stands for what? For the peptide bond, because this thing here, this circle thing, is called a peptide bond, which is a type of covalent bond, and a dipeptide, di for 2, because it joins 2 amino acids, and peptide for the peptide bond, okay? Hope that helps you remember. Exactly. Now, this is the C-terminus, the carboxyl group of this amino acid at the right, and this is going to be the N-terminus, the amino group of on the left of this dipeptide. Now, if you repeat this process more times, even just once, you now have a polypeptide because it's for 2 or more peptide bonds. Okay, so we have 1 peptide bond here. If we added more amino acids, we would have more. Okay, and that's why it's poly. I believe I mentioned essential and non-essential amino acids. Okay, so having a look, we can see that essential amino acids must be consumed by food. Why? Because they cannot be synthesized or produced by the ribosomes in your cells. You can't make it. That's why you need to consume it. Non-essential amino acids are synthesized by the body, and so you don't necessarily need to consume them. Now, transcription, this is relevant, okay, because how do we synthesize these non-essential amino acids? Well, first step is transcription. This is when DNA is transcribed into mRNA, sorry. I have a video on this as well. I believe it's the point something. Now this is followed by translation, where the mRNA is translated into amino acids. We're going to have a look at that in a second, because these are all the different amino acids. The PHE is one, the LEU, we can see LAA, okay? So these things at the right are the amino acids, and these are the codons that they code for. So PHE codes for UUU and UUC, and PRO codes for CCU, CCC, CCA, etc. Okay. Now, there's an infinite variety of possible amino acid or peptide chains. Okay, as you can see, there's so many different ones. So there's an infinite variety of combinations you can make. Now, the genetic code is degenerate. What does this mean? Okay, this is the genetic code here. What does this mean? This means that some amino acids are coded for by multiple codons. Okay. Now, this means that, for example, LEU is coded by CUU, CUC, CUA, and CUG. So all of these code for LEU. That's why this is degenerate. For example, for ARG or ARG, all of these code for it. Pretty simple. Now, this allows for silent mutations, which is essentially when you change one of the codons. For example, you replace UUU with UUC. You do this replacement, but it doesn't change which amino acid is produced because it's still PHE. Understand? Any questions can go in the comments as well. Now, the genetic code is also universal, which basically means that all organisms in the world have the same 20 amino acids. A protein is made up of at least one polypeptide chain. Okay. Now, if it's made of only one polypeptide chain, the amino acids are going to interact with each other, and so this protein is folded into what's called a functional protein. Now, if we have two or more polypeptide chains, the polypeptides interact with one another, and it does not lead to a functional protein. Now, let's look at different examples of polypeptides, starting with lysozymes, the zyme for enzyme, okay, that is present in tears and saliva, and it's going to disrupt the cell wall of certain bacteria, so it's responsible for your immune system. Okay, it's going to help your immune system. It's present in phagocytes, I believe, so it's very important. We can see alpha neurotoxins are found in snake venom. They disrupt the nervous system. Glucagon is a hormone. You'll see it in other topics. It's secreted by the pancreas, and it stimulates glycogenolysis, which is breaking down glycogen into glucose, and gluconeogenesis, which is producing glucose from non-carbohydrate sources such as fats. Okay, and this happens in the liver. Again, this is linked to other topics, so if you don't understand what I'm saying, that's fine if you haven't studied the other topic. Myoglobin is an oxygen-binding protein, and it facilitates the storage and release of oxygen in muscle tissue. That's all there is to it. Now, this links to the topic of denaturation, enzymes and stuff, which I hate because I've done a million times, so didn't even bother animating it. Let me explain this real quick. Now, the structure of the proteins are going to determine their function. So any change in this structure can lead to a loss of activity or a wrong activity. Now, denaturation is the alteration of the structure, and so it loses the function, as we said before. We know that enzymes are proteins, and so this is basically the same as how it applies to enzymes. Main causes: pH changes, temperature changes. They each have an optimal pH and an optimal temperature. Extreme pH alters protein charge. Okay, let me underline that. Now we actually know why it alters the protein structure, because it alters its charge, and this affects the solubility and shape of the structure. Now this can cause irreversible damage. An example of this is pepsin in the stomach, which works in acidic conditions, and it can become inactive in alkaline pHs. Effective temperature. High temperature breaks the hydrogen bonds. Okay, hydrogen bonds, and so the protein unfolds. Most human proteins work best at 37 degrees Celsius. This is a fact which can come up as a multiple choice, so have it in mind. 37 degrees Celsius. Heat-loving organisms have proteins adapted to high temperatures, and low temperature can affect structure slightly, but less than high temperature, so high temperature is apparently worse.